Hi, good morning. Thanks to everyone for being here and for being so excited about learning about biosimilars. It's a little bit of a rabbit hole topic, and we often don't get as much enthusiasm as we should. So I'm going to do my best to go through this. I might stand at the podium, but I might try sitting here first or standing here. I don't know. You guys, if you can't see me or hear me, let me know. And if I start to talk too quickly, shout, wave your arms. I have a tendency to do that, so I'll try to be mindful. I have a whole bunch of topics to go through, and then we're going to have some time at the end for questions. So definitely prepare your questions. There's a lot of material here, but I'm going to try to hit the big highlights of what we are doing, starting with FDA's role in biosimilars and therapeutic biologics. So, there's a lot of talk right now about balancing innovation and competition. The FDA's role is an important one. This is a multi-stakeholder endeavor, obviously, but we recognize that we have a very important role in helping to ensure a robust marketplace and timely competition in these products. And as part of that, what my office is really focused on is playing a critical role in facilitating increased access to biosimilars. And that includes, when I talk about biosimilars, I'm also talking about interchangeable biosimilars, which we'll talk a little bit more in a bit about what that all means. But at the end of the day, increasing treatment options and creating a robust marketplace for biosimilars, we hope will improve patient access to biological products. As part of that, last summer we rolled out the Biosimilars Action Plan, which is an agency-wide effort to enhance sort of efficiency, clarity, communication development, and generally support market competition by finding places where we think there might be gaming in the market, reducing gaming, and sort of stymieing attempts to unfairly delay competition. So the Biosimilars Action Plan is a good read if you're interested in this subject matter area. And we've made a lot of progress. I'll talk a little bit about, at the end about where we're headed, but for now we'll touch on some of the things. We call them BAP deliverables. The BAP is the Biosimilars Action Plan. And a lot of the deliverables in here are about enhancing clarity, stimulating development of these products. So we've put out a series of question and answer guidance documents, several guidance documents dealing with the BPCI Act transition, which we'll talk about in a little bit more detail, non-proprietary naming guidance. We had put out guidance. We've recently updated that guidance. We'll talk about that more in depth. We held a Part 15 hearing on biosimilar and interchangeable insulin specifically for that subject matter area, and we're doing a lot of work there. Considerations in demonstrating interchangeability, which came out a few weeks ago. It's a final guidance that talks about some of the considerations in designing development programs for interchangeables. And just last week, development of therapeutic protein biosimilars, comparative analytical assessment, and other quality-related considerations. And we'll talk a little bit more about some of those and what they mean and what they contain. So where are we as a program? Well, as of May 1st, we had approved 19 biosimilars. None of them were approved as interchangeable. They were all approved as biosimilars, referencing 9 different reference products. Now, a reference product is defined under our statute as a single 351A biological product that is against which a 351K biological product is evaluated. So when we're talking about biosimilars, we're talking about applications submitted under Section 351K of the Public Health Service Act. When we're talking about originator biologics, we're talking about applications that are submitted under Section 351A of the Public Health Service Act. As of May 1st, we also had 78 programs enrolled in what's in our BPD program, Biosimilar Development Program, which is the user fee program that governs the engagement between the FDA and potential applicants. And we go fairly in depth in the BPD programs to work with sponsors on a product-specific basis to discuss development of their proposed products. So as of May 1st, we had 78 BPD programs. And there have been 29 publicly announced planned 351K submissions. I actually read something last week about a publicly announced 351K submission for an interchangeable product, which was, I think, new to me. But this is all, this is all a very new program. So let's go through the regulatory background, talk about what the program looks like. So as I mentioned, an originator biologic we call them standalones. They're submitted under Section 351A of the Public Health Service Act, and they're evaluated against a standard of safety, purity, and potency, which is generally considered to be analogous to the safety and effectiveness standard under which NDA products are evaluated. In 2010, the BPCI Act created an abbreviated licensure pathway for biological products that are shown to be biosimilar to or interchangeable with an FDA licensed reference product, right? I mentioned that the reference product is the 351A product against which the K product is evaluated. That's an important piece of our statute. The abbreviated licensure pathway means that a biologic that's biosimilar to or interchangeable with a reference product can rely in part on FDA's determination of safety, purity, and potency for the reference product. The idea being that that can streamline the amount of clinical trial development that has to happen for the program. The button stopped working. There. As a result, generally, biosimilar manufacturers won't need to conduct as many clinical trials. That can reduce costs considerably. It can also speed the process. It's not a change in the approval standard. It's important to remember that. So in 2015, we approved Zarxio, which was the first FDA-approved biosimilar, and we've been really building the program from there. I want to talk a little bit about the standards and what all these words mean. So biosimilar or biosimilarity means that the biological product is highly similar to the reference product, notwithstanding minor differences in clinically inactive components. That's the statutory definition. It means that there are no clinically meaningful differences between the biological product and the reference product in terms of safety, purity, and potency. So again, the goal here is to establish biosimilarity, not to redo all the clinical trials, not to reestablish safety, purity, and potency, or safety and effectiveness, but instead to establish a comparative relationship between the biosimilar and the reference product, and that's how the pathway works. It's similar to the generics pathway in the small molecule space, but has some differences based on the nature of biological products. How do you demonstrate biosimilarity? So the statute actually lays out the factors that have to be included in a 351K application to demonstrate biosimilarity. The biosimilar or the proposed biosimilar has to utilize the same mechanism of action to the extent the mechanism of action is known. Sometimes with biologics, it can be very difficult to characterize the molecules, and so we end up with unknown components. But to the extent that we know, the mechanism of action has to be the same. The conditions of use that are proposed in the labeling for the biosimilar have to be previously approved for the reference product. So you can't come in with a biosimilar application with a new condition of use for which the reference product hasn't been approved. They have to have the same route of administration, dosage, form, and strength, and the facility in which they're manufactured have to meet standards that are established to ensure continued safety, purity, and potency. Now, biosimilar and interchangeable products can be used in patients who have been treated with the reference product or who have not been treated with the reference product. It's not a previous requirement in any way that the patient who's being prescribed the biosimilar product have been or have not been previously treated with the reference product. And it's also important to realize that a biosimilar can be approved for all of the conditions of use that a reference product is labeled for, or for a subset, or for less than all of the conditions of use. So there is some room for deviation there. A little bit about the science behind a demonstration of biosimilarity. It's a totality of the evidence approach, right? So we talk a lot about stepwise development, but we ultimately are approving a biosimilar based on the totality of the evidence. Data is generally from analytical studies, sometimes animal studies, and clinical studies. We'll talk a little bit more about some of those things in a minute. Generally, the nature and scope of the clinical portion of the development program is going to depend on the extent of residual uncertainty about the proposed product after the comparative analysis is done. So the first step is going to really inform how the rest of the program works. We recently published this comparative analytical assessment guidance just last week. It talks about our recommendations for the design and evaluation of comparative analytical studies. It's been a big outstanding commitment. We've been working really hard to get it out. We're really happy about it. It's fairly complex, but in general, it includes a stepwise approach to demonstrating biosimilarity. So it lays out sort of how you go about detecting and characterizing differences between the proposed biosimilar on the one hand, the reference product on the other hand, how you then assess the differences between those two things to understand the impact on clinical performance of the proposed product relative to the reference product, and then how you can design the rest of your development program, how much animal data might be needed, how many clinical studies or clinical data might be needed, and how those additional studies might be structured. So there's a little bit here about areas where we tried to provide some additional clarity and flexibility for developers in terms of how to approach evaluating product structure and function, how to analyze comparative data, potential alternative approaches to the extent that they're appropriate and desired, and some of the important aspects of the CMC portion of the marketing application. The chemistry and manufacturing pieces of a biosimilar application tend to be very important because, again, the basis of the sort of foundational piece of a demonstration of biosimilarity is the comparison between the two products, which has a lot to do with the chemistry and manufacturing. So the other big bucket, right, we have the 351K biosimilars, and then we have the 351K interchangeables. Interchangeability is not a different standard, but it means that the biosimilar can be substituted at the pharmacy level without the intervention of a healthcare provider. So a biosimilar, a healthcare provider is going to write a prescription for that specific biosimilar, and interchangeable will operate more like a generic drug. At the pharmacy, the pharmacist will automatically substitute those products. It is sort of dependent on state law. We're not going to go into that because that would be like a whole other 40-minute presentation. But at bottom, this is really, interchangeability is really about pharmacy level substitution. It can be complicated to demonstrate interchangeability. We put out a final guidance 2 weeks ago that talks about how interchangeability should be demonstrated, or considerations that should influence programs that are intended to demonstrate interchangeability. Again, though, it's a stepwise approach, a totality of the evidence approach. We're looking at the whole submission to determine whether it meets the standard for interchangeability. What is the standard for interchangeability? Well, it's also in the statute. There are three prongs to demonstrating interchangeability. So the first one, the proposed interchangeable has to be biosimilar to the reference product. That's the same as what we just talked about, right? So if the product has previously been licensed as a biosimilar, the interchangeability application can rely on that finding, can reference that finding. 2, the application has to demonstrate that the proposed interchangeable is expected to produce the same result in any given patient. This is typically not going to involve additional clinical studies other than those necessary to support prongs 1 and 3. And this is a big one with a lot of words, but at the end of the day, this is about for products that patients will be switched back and forth between, right? Remember, you're being substituted at the pharmacy level, so you may be going back and forth between the interchangeable and the reference product. There needs to be a demonstration that that switching or alternating between interchangeable product and reference product won't increase the risk in terms of either safety or effectiveness. And generally, that will be demonstrated by something we call a switching study. The interchangeability final guidance talks a lot about switching studies. What are we looking for? How should they be developed? What kinds of study designs might be appropriate? And generally, that switching studies are not going to be needed unless it's a product that's going potentially to be switched or alternated and given to an individual more than once. There's also a lot of discussion about non-US comparators. So when you're developing your interchangeable product and you have to do a switching study, do you have to be doing that switching study using a US licensed reference product, or can a foreign comparator be used? Some of the considerations there are outlined in that guidance as well. And there's a lot of discussion about post-marketing data. That is, what role does post-marketing data or actual patient use information play in determining interchangeability? And this is sort of, the guidance clarifies a little bit where we think it might be useful to have post-marketing data be part of an application package, but ultimately it's about the types of therapies that are being looked at and the potential for certain kinds of immunogenetic adverse events to happen. Okay, interchangeable versus biosimilar. Basically, I just wanted to really highlight this because I think it's a point of some confusion. There are different regulatory structures for these two types of products that reflect the different scientific considerations for certain types of products. It's not a different standard, right? It doesn't mean that there are differences in safety or effectiveness. It means that, for example, one type of molecule has a history of triggering dangerous immune responses, and so we need a different kind of data to be comfortable with those products for approval. There are also different business considerations that influence applicants' decisions about which types of licensure they might be looking to seek. So you would have, as a business matter, a different case potentially for a product that is administered to inpatients by the physician versus a product that would be dispensed at the pharmacy. Okay. Is everyone still excited to be here? All those words later. Still, okay, still smiling. Good. We're going to talk a little bit about the transition. People call it the transition because it's coming. But essentially, there are certain types of biological products that traditionally have been regulated under the Food, Drug, and Cosmetic Act. So under NDAs, right? When Congress passed the BPCI Act, it also included a provision that said on March 23rd, 2020, those NDAs are going to be deemed to be BLAs. So there's a whole list of products that are going to move from regulation under the Federal Food, Drug, and Cosmetic Act to regulation under the Public Health Service Act. And instead of having NDAs, those products are going to have BLAs. And FDA, as we've announced in FDA guidance, that those 351 BLAs are going to be 351A BLAs, which means that applicants can then come in and seek to be biosimilar to or interchangeable with those products. We've issued several guidances talking about our thinking on these deemed BLAs, what they will mean. We're working a lot with applicants to minimize the effect of the transition. It's been 10 years coming, so we've known about it for a while. We've been thinking about it for a while. Sponsors have been taking it into consideration for a while. We do think at this point that our performance goal dates and timelines for review and the way that we've worked through it with sponsors have made it very unlikely that any proposed or pending applications would be affected by the transition. And we are going to be able to maintain our goal dates for completion of review of pending supplements, which means the date by which, under our user fee commitments, we are supposed to have completed review of the products, we expect to be able to maintain. There will be some differences after the transition, right? So there are some differences between the way that we label, for example, BLAs and NDAs. Carton and container labeling have some small differences. Certain required content in the prescribing information will be different. We have announced our intention to not take enforcement action based on those differences until March 2025. So there will be a 5-year period of this compliance policy, which really we think will minimize the disruption and burden associated with making the changes that sponsors holders of those applications will need to make. Okay, Safety and monitoring. I know everyone's really excited to talk about our non-proprietary naming policy. That conversation, I think, starts by a discussion of the safety and monitoring of biological products. So all drugs have risks and benefits, right? Biosimilars have side effects. We expect those to be the same as the side effects and the adverse events associated with the reference product, but we monitor for those adverse events across the board. So as part of our review, FDA assesses the manufacturing processes and the strategies to control product variations. Those strategies are supposed to help ensure that manufacturers produce biological products that are consistent, right? That perform consistently. But at the same time, there has to be robust post-marketing safety monitoring. It's an important component in ensuring the ongoing safety and effectiveness of all of the products, including biosimilar products. So as part of our implementation of the BPCI Act, FDA requested public comments in public meetings held in 2010 and 2012 on how to develop a framework for safe use and optimal pharmacovigilance for biosimilar and interchangeable products, including the role of the name of the product. And those meetings and all of that thinking informed some earlier drafts, some earlier guidance. So in August 2015, we published the first draft guidance on non-proprietary naming. We finalized the guidance in January of 2017. And then more recently, we published an updated draft guidance, which we'll talk a little bit more about what exactly that says. But at the end of the day, it starts with the conversation about safety and monitoring, right? What are the core objectives of the naming convention? Transparency, right? It needs to allow identification of products for safe use and for pharmacovigilance. You don't want to have a situation at the end of the day where something goes wrong with a drug product and the investigators can't figure out what the patient took. Patients and practitioners both want FDA to have the tools available to perform product-specific pharmacovigilance, right? And it's important that the public be confident in the monitoring process and that confidence be able to facilitate market uptake. So how do we name biological products? There's a proprietary name, right? The proprietary name is generally trademarked and registered for private use. And then there's the proper name, which is also known as the non-proprietary name, and that's what we're going to focus on here. That name generally reflects the scientific characteristics of the product, the chemical structure, the pharmacological properties, and FDA designates a proper name for use upon each package of the biological product. So with all of the goals in mind, we developed a naming convention that we think is necessary to ensure safe use and pharmacovigilance for these products. What is that naming convention? So the proper name is the core name plus a designated suffix. It's a 4-letter suffix. And the Levna, who's going to speak after me, is going to talk a lot about all of the work and energy that goes into this process of designating the names. But I'll just say that the suffix will be given to and applied to originator products, biosimilars, and interchangeables with some limited exceptions. We think that over time, these suffixes will increasingly become the norm for biological products, right? So now it seems new. The whole program is new. But over time, as all of the products have suffixes, as originator products have suffixes and biosimilar products, it should become the norm. And at the end of the day, it's about providing transparency and predictability and ensuring safe use and pharmacovigilance. Okay, a little bit more on safe use. And this is largely discussed in the guidance that I mentioned, but I'll just hit on some of the high points because I'm going to try to save enough time for questions. And it's amazing how quickly the clock runs out while you're up here. But the ability to clearly identify biological products can help ensure safe use, facilitate accurate prescribing and dispensing. And in clinical practice, it's important to be able to distinguish among originator products, related products, and biosimilars, right? So for example, I mentioned earlier, a biosimilar product can be licensed for the same conditions of use or not. it can be licensed for fewer than all of the uses or routes of administration for which the reference product is licensed. It's important that a practitioner be able to distinguish across products if you're talking about a use for which the biosimilar is not licensed. So that's a good example of the sort of kind of safe use considerations that we're thinking of. On pharmacovigilance, we have a lot of both active and passive pharmacovigilance systems, and a lot of them have really useful abilities, but they do have a limited ability to track an event to a specific manufacturer for a biological product, right? So again, you know, if an investigator is in a patient's home looking in their medicine cabinet, the suffix will help determine exactly where the product came from and was manufactured. So our most recent update to the guidance, which was published in March 2018, sort of stuck with the position that applying a suffix is necessary for safe use, to ensure safe use in pharmacovigilance, but also said that we don't think it's necessary for products that are already licensed. The costs associated and the burden associated with going back for a product that already has a license, where the materials are already printed and the name has already been marketed. We got a lot of comments about the extent of the burden and cost associated with that. And we ultimately, in the updated draft guidance, took the position that already licensed products would not need to have suffixes to achieve the goals of safe use and pharmacovigilance. Whoops. And so that means that also, in addition to previously licensed BLAs, also the transition products, the products that are transitioned from NDAs to BLAs next March, will not be getting suffixes under the guidance. So that's my 2-minute overview of non-proprietary naming. Okay, where are we going? Here are some things that we're currently working on. We're drafting additional guidance to provide clarity to biosimilar applicants who are looking to develop labeling when they're applying for licensure of fewer than all conditions of use. Draft guidance on product presentations for interchangeability. An enhanced version of the Purple Book. Have you guys talked about the Purple Book? Do you know what it is? Do you know what the Orange Book is? Okay, so the Orange Book is for generics. The Purple Book is for biologics. It lists our biosimilars and interchangeables. And it's sort of like the sad cousin of the Orange Book. It's basically, at this point, it's a spreadsheet, but we're going to make it great. So we're working really hard on that. I think it'll be really helpful. People are really looking forward to it. And the last thing I want to mention, 2 things. We're also working really hard on a regulatory modernization. We're overhauling the Part 600 regs that govern the content and filing of BLAs. So stay tuned. I think it'll be a while before that regulation is finalized and becomes effective, but when it does, there will be new things to learn and talk about. And we have a whole new, look, there's a whole slide on the Enhanced Purple Book, and it's purple. That's how excited we are about it. Although there's a lot of discussion in my office about what color purple the Purple Book should be. Should it be more lilac, more magenta? Happy to take feedback. I picked this color for this slide. You can tell which camp I'm in. So the biologics regulatory modernization, as I mentioned, and we're doing a lot of education and outreach. We've spent a lot of time and energy over the last few years trying to educate healthcare providers about what biosimilars are, what they mean, how they should be how healthcare providers should be interacting with these products. We're also, these are some of the materials that we've developed, and now we're in the process of launching and working on a campaign of education about these products that's really targeted at patients and that will hopefully have more user-friendly materials. I'm sure you noticed in the last 29 minutes, I'm really bad at math, that's why I'm a lawyer, that these things can be really difficult to talk about in an efficient way and in a user-friendly way. So it's a challenge, but we're tackling the preparation of more patient-focused educational materials. And I managed to leave 11 and a half minutes for questions. Does that mean we turn the mics on? You thought that it was going to take me longer than that to get through my slides.
So all of you remember from yesterday, if you have questions, there I'm on. If you have questions here in the room, please come on up to the microphone. Those of you online, thank you for the questions that have been submitted thus far. If you want to submit questions, the lower right-hand corner, that Q&A pod, just go ahead and type those questions in, and we will be reviewing them as they come in. But let's go ahead and start here in the room. Microphone number 1, please. Up. He's getting there.
Got it.
Yep.
I'm Sonia Lohr. Can you comment on the current status of the 4-digit suffix for new vaccines?
So, yeah, so did everybody hear the question?
Yep, please repeat.
Repeat, see?
Yeah,
Ye of little faith. The question was where we are on the 4-digit suffix for vaccines. So one of the things that was mentioned in the March 2018 updated draft guidance was that we are reconsidering the suffix for vaccines and whether or not it's necessary to ensure safe use and pharmacovigilance in the same way that it is for other products. There are a lot of vaccine-specific monitoring programs. I think the example in the guidance is the, I'm going to forget exactly what it's called, but there's a program for reimbursement for vaccine events, and that requires certain kinds of tracking that is unique. And so we are in the process of evaluating whether the suffix is necessary in light of those programs or whether there's some redundancy there. We will eventually publish additional guidance on it. We don't have an answer at this point. But it's a great question.
All right, microphone number 2, please. Go ahead.
Hi. So what happens if you already have a non-proprietary name assigned by the INN or USAN? So in that case, will the FDA adopt the same generic name, or will they assign a new name altogether?
So go ahead.
No, I'm not going to repeat it. You can repeat it if you want. I don't repeat questions because they go over my head.
The question is, and I might ask you to repeat your question too, but I think your question is if you already have an assigned non-proprietary name.
Yep. So you can get an assigned non-proprietary name from international organizations such as the INN or in the US, USAN. And so if you've already applied for a non-proprietary designation and you've been granted one, then when you submit your BLA, will the FDA adopt the same name, or will they assign a new one?
I'm going to recommend that Lubna answer that question when she comes up here in a little bit.
Okay.
It's a good question. I have theories on it, but rather than bungle them, I'm going to defer that one. She's nodding, so I think she knows the answer.
All right. Thank you.
And Lubna is our next speaker, so you'll get that answer shortly. All right, and any other questions in the room? We've got about 7 minutes. Come on up to the mic, but let's go ahead and take our first online question, please.
If the original product had some limitations in terms of efficacy or side effects that could not be improved due to lack of technology, should one try to address such issues while developing biosimilars, or simply stick to the matching the reference compound in terms of efficacy and side effects?
You guys asked really good questions. So the question is, if there may have been some issue with the reference product during its development program, is that something that a biosimilar developer should try to overcome, or should the focus really be on developing a comparative product? You know, we're very aware of this question. It's an open question and something we think about a lot. I think, you know, the standard is highly similar with no clinically meaningful differences, and so ultimately it would be a question, I think, of whether there are clinically meaningful differences between the proposed biosimilar and the reference product. But we're certainly mindful that science marches on, and I think we try to adopt flexible enough approaches that as science progresses, we can work with sponsors in a product-specific way to talk about those questions.
Okay. And again, if there's any other questions in the room, come on up to the microphone. Otherwise, Ray, let's go with another online question, please.
Could you briefly speak to the different fees needed for the applications of biosimilars and if there's any difference between that and interchangeables?
So there should not be a difference in fees. Sorry, the question was whether there's a difference in user fees between an application for a biosimilar and an application for an interchangeable. We don't expect that there will be a difference in fees for BPD for the development of a 351K application or the submission of that application based on whether it's seeking biosimilarity or interchangeability. That said, if a product is already approved as a biosimilar, has already paid the fees associated with that, and is now seeking to become interchangeable, that may be a different type of application that would have different fees associated with it.
Okay. Got another one back there, Ray?
Can an interchangeable application be done in parallel to the biosimilar application?
So that's a great question. We're actually working on some guidance right now that will have additional information about the administrative issues around submitting applications for biosimilarity and interchangeability. At this point, we've not developed guidance on the precise question. I mean, I think an application could come in and be reviewed for interchangeability. Part of the determination of whether a product can be licensed as an interchangeable involves a finding of whether there's been an appropriate demonstration of biosimilarity. So one is sort of inherent in the other. But it's a good question as an administrative matter, and we'll certainly think about ways to address that.
All right. And Eva did let me know that she's got to leave for a flight, so she won't be able to do the Q&A at the end of the day. So if you have any questions here in the room, now is the time to come up the mic. Another online question for me, Ray?
And this is dealing with smaller polypeptides. Do smaller polypeptides fall into the definition of analogous products?
I'm not sure what is meant by, so the question was whether smaller polypeptides fit into the definition of analogous products, and I'm not sure what's meant by analogous to what, but since maybe the person who asked the question can
yeah, if whoever typed that question in the Q&A pod
little bit more
can type some clarity in about what you mean by analogous, we can circle back around to it, possibly. Otherwise, Ray, let's go with another one.
Just to revisit that one again, they did, assuming it's correct, get some information from a guidance. It says BPCI Act deemed to be a license provision guidance biologic product definition, and they have ... protein except any chemically synthesized polypeptide or analogous product. And
right.
So that was the sentence that they were referring to.
So can you go back and repeat the original question then? I'm trying to piece it all together.
Do smaller polypeptides fall in the definition of analogous product?
I now understand the question. I'm not sure that I have a satisfying response, right? So the BPCI Act, the way that biological product is defined, it includes proteins, and then there's a parenthetical in the definition that says except any chemically synthesized polypeptide. And the question, I think, is whether a smaller polypeptide or analogous product would fit within that little piece of the carve-out of the definition of protein within the definition of biological product in the Act. And I don't know if, I'm trying to think about what we have publicly said about that, and I'm not sure that at this point there's an answer. But typically, the smaller amino acid chains will fall out of the definition because of their smaller size. I'm not sure that that really answers the question, but I can certainly think about it a little bit more and maybe provide some feedback.
And again, for everybody online and here in the room, if there are questions that we don't have time to get into, CDER SBIA at fda.hhs.gov. You can send an email there. They will route it to the right place and get you an answer. We've got time for one more online question.
Are there any differences in the review timelines regarding original BLAs and biosimilars or interchangeables?
So I believe that and I really should know the answer to this question with more certainty than I do. I believe that the answer is no, but the original, the 351A applications will be governed by the PDUFA timelines for review of an application, and under PDUFA, I think it's a 10-month timeline. Under BSUFA, which governs the 351K applications, I think it's also a 10-month timeline.
Okay. And that's all the time we have for, oh, okay, sneak one in real quick. You got 5 seconds. Oh, he's turning right back on now. Okay.
Can you comment on the Part 600 updates and whether or not there will be a specific provision added for referencing master files? But comment overall and then about master files and whether or not there will be a specific provision added.
So the Part 600 modernization, which is referenced in the map, is really about finding places where there is a lack of clarity about biosimilarity, right? The Part 600s were written before the 351K pathway existed, and so there are places where they really aren't contemplated, where this other type of abbreviated licensure pathway really isn't contemplated in the regs. And the reg modernization is really about finding those places and patching those holes where we can provide clarity and certainty for biosimilar applicants. It's not, and we are, as part of that process, reviewing all of the Part 600 regs and looking at places where we can get the most bang for the regulatory buck, for lack of a better way to say it. At this point, I think the DMF questions are being handled separately.
Okay. Please help me thank Eva for coming and talking with us this morning.